Morning, everybody. Welcome to our first snow day of the year. Hopefully, our last snow day of the year, but no promises on that. I am coming to you live from my house where you may hear some tweets in the background. That is Foster, our lovebird, um, as well as you may hear some dogs at some point. So bear with me. That's Foster. Um, Today, I want to probably finish up the notes um, for chapter 16. And if you recall, before we went on break, I had talked about doing biotech. After we came back, I said we're not going to do that during the cell cycle, and I said we'd do that when we got back. And given how quickly we're going through these notes, I think we're going to do biotech too. That's not too much. I do have notes on that that I can give you on Tuesday, or I can share them online if we don't have school on Tuesday. But let's start by finishing up chapter 16, and then we can talk about that. So we left off yesterday. We'd finished with the leading and lagging strand and DNA replication, and that's really all there is to that. That's really what you need to know, and please make sure that you understand that because that will be the bulk of your test that you'll get next Friday. But there's a little bit more we need to follow up with and talk about. So obviously, when DNA makes a copy of itself, it's very important that it does it correctly. And there are ways to deal with that if something gets messed up. So there's proofreading that happens. And remember, we have this molecule called DNA polymerase. And in the textbook and other places, it may talk about DNA polymerase 1, 2, and 3. And we're not interested in differentiating between those three different types of polymerases. We just have to know them collectively as DNA polymerases. So there is one kind of DNA polymerase whose job it is to, for example, lay down nucleotides, to make put in DNA nucleotides. There's another DNA polymerase whose job it is to proofread the DNA, and in the case of errors, to change that up and make sure that that is done correctly if there's an error. So in this case, we call this a mismatch error or repair, where the DNA is not matched with its correct nucleotide. And that can happen from a variety of environmental factors. It could happen from different chemicals or agents in the environment, like cigarette smoke. Cigarette smoke has lots and lots of different carcinogens in it. There isn't just nicotine. There's other things that also cause cancer that are in cigarette smoke. It's pretty nasty, as well as obviously just cigarettes in general, as well as x-rays. Gasoline would be another example. I know it's not listed on here, but benzene is another one, asbestos. All of these can lead to the DNA getting damaged. So it leads to what we call just a spontaneous change. So something happens in that ACTG sequence is messed up. And so when you go to lay down that new DNA nucleotide sequence, for whatever reason, it doesn't match with the right nucleotide. And so when that happens, you need to repair that. And that is called nucleotide excision repair. So basically, all that means is you're taking the nucleotide and you're cutting it out. So excision means to cut it out. And you do that with a group of enzymes called nucleases. And those nucleases are responsible for taking out the messed up part. And so that's shown in this series of drawings in this next picture. Here's the first one. And again, we don't want to have all that. So let's say, nope, now it doesn't want to get rid of it at all. That's really annoying. Let's see if I can clear the screen. Nope, it doesn't want to clear the screen either. Okay, so sorry about that. I'm going to, while we're talking about this, I am going to go ahead and duplicate this tab. And then I'm going to go back to the old one. I'm going to talk about this, and hopefully that will load in a decent amount of time, and then we can go to that screen and we won't have these pictures on here. Sorry about that. So here we have a piece of DNA that's messed up. And so you can see the mess up It's not fitting correctly in that location. And then the nuclease is going to come along, and its job is just going to be to cut out that piece of messed up DNA. Once the nuclease takes it out, our old friend DNA polymerase will come along, and it will add on DNA nucleotides just like it does in the leading and the lagging strand. But there's going to be a little gap here that is not connected. That's between the phosphate of one nucleotide and the sugar of another. We call that the phosphodiester or phosphate sugar backbone. And so even though DNA polymerase lays on those DNA molecules, those DNA nucleotides, we have to have one more step, and that one more step is going to be you to use this thing called DNA ligase. And DNA ligase is kind of, think about that as the glue. That's the thing that's going to tie those two pieces together. So it's going to take that piece here, and it's going to it's going to put in a bond, a phosphodiester bond between that phosphate of one nucleotide and the sugar of the next. So now it's all ready to go and ready to rumble. Okay. Now, obviously, that doesn't always work, and sometimes these errors go on, and whether or not they are of consequence is kind of random. And I talk about people, even young people, getting cancer, and it's kind of random whether or not that happens. As you do age, those errors, those places where the nucleases haven't been able to repair the DNA, that's going to increase. So you do see in older people an increase in cancer because they just have more. They have more replication events. Their DNA has copied more times. And because it's copied more times, there's been more places where there's room for error, and there's places where the error has occurred. And so accumulation of the errors that could lead to, remember we talked about P53 and we talked about oncogenes. And so if those errors occur in those particular genes, then we could either end up with a gene that would not be produced, P53, for example, that would not be produced to stop the DNA from going forward and producing more bad copies. Or the other thing is, remember, we had oncogenes as well. So oncogenes are the ones that turn on the cell cycle, and normally they get turned off because you only want the genes to be turned on, and you don't want that to be produced when there's only a need for example, if you cut yourself. But if an oncogene gets mutated into the on position, then what will end up happening is the cell cycle will just continue and continue and continue, and then you'll get this ball of cells that will be cancerous. But that happens typically as you get older, but it wouldn't necessarily have to be as you get older. Okay, so I'm going to switch over to two things. First of all, I'm going to go into a different room so we can hear each other. And then now I've got my new slideshow, and I'm assuming that, again, as I make new notes on here, it's not going to work either. So I'm going to go back to this one. Okay, so now I want to talk about, I wish we were in class right now because I think this next little piece that we're going to talk about is really cool. And we'll talk about it again when we get back in the classroom. It's assuming this actually works. There we go. Okay, so this is not what I think is cool. It's the next slide, but let's talk about this one first. So there is an impact on evolution because of the fact that there are altered DNA sequences. Not all of this is fixed. So we know that there's cancer, and we know that proofreading doesn't always work, right? The repair rate is the error rate, I should say, after proofreading is pretty low, but it's not zero, okay? It's not 100%. There are some things that don't get fixed. And in some cases, that might not be consequential in terms of the organism's lifespan. So maybe I have some particular change in my DNA, but it doesn't actually change to the point where I'm affected and I die from it. And I pass that particular change in my DNA on to the next generation. Okay. That is important. It's significant because what happens is it leads to variation in DNA from person to person over the generations. And so that could be harmful, but it could also be helpful. Now, remember, when we talk about these mutations, we talk about these changes, these permanent changes in the DNA. Say my DNA gets mutated. All we're talking about when we talk about a mutation is just a change in the DNA sequence. That's a mutation. But let's say that isn't harmful. I still have children and I pass that DNA sequence on to my kids. That mutation is genetic variation that could potentially lead to, and this is talking about, very, very far down the road, but it could ultimately lead to a new species. Okay. And it could just be a very, very subtle thing, like a new species in that species is able to, withstand much colder temperatures than someone who doesn't have that particular mutation. We're not talking about having a mutation that suddenly I have wings and I can fly. Okay, we're talking about very subtle things. And we've seen this happen, and we'll talk more about this when we get to evolution. And we've seen evidence where one gene has served a particular role, and then the next thing you know, that gene gets mutated, but it gets mutated in a way that actually it still produces a protein and it still produces some significant outcome, but it's different from what it used to be. And we've seen this in particular cases. The one that's coming to mind right now is in some fish have what we call an antifreeze gene, meaning that they can swim at levels that are very close to freezing. And that is significant because they're able to live in much colder environments than other fish. And there's evidence to show that gene didn't exist for many years, and the sequence of part of that gene suggests that gene was a different gene at one point and led to a different protein, hundreds of thousands of years ago. But because of some mutations, those mutations now cause that gene to produce a different protein, and that protein has different effects. And we'll come back to that, and we'll talk more about that when we get to evolution, because it is a pretty cool thing. But what I really wanted to talk about was not, see, I knew this was going to happen. And yep, can't clear the screen. Awesome. Oh, it's a snow day for sure. Okay, going to go to our third PowerPoint here. And scroll down to where we are. I think it's that one. There we go. And I'm going to make another copy so that we'll have some duplications for later. How about we just make another one, because this is going to happen again. And again. It's so silly. Please tell me I didn't just X out of that. Okay, I didn't. Good. So we talked about how there's a leading and there's a lagging strand. And we talked about how that is because of the fact that the DNA polymerase is working towards the replication fork in one case and away from it in the other. And we also talked about how DNA polymerase is a little bit needy in that it cannot add on DNA nucleotides unless it has a primer attached to it. Now, that's awesome. I mean, maybe not awesome, and you may think, and I know several of you have been like, this is wiggity whack that this happens, and it is weird, but it is the way our DNA replicates. It has to have that kind of crutch or that structure, that something physical that it can attach to. And the problem with that is that DNA polymerase then has problems when it gets to the very end of the DNA. So this is a problem for eukaryotic chromosomes. And it's only a problem for eukaryotic chromosomes. And here I go with my underlining again, that will probably be an issue. It's not going to be a problem for prokaryotes to have circular DNA. And that is because of the fact we've got this leading and we have this lagging strand. So what I'm going to talk about next is only going to apply to linear chromosomes, and linear chromosomes are only found in our eukaryotic chromosomes like ourselves. It's not going to be found in prokaryotes or bacteria. So when I'm talking about linear, I'm talking about this. Let me make this off to the side so that it doesn't. Okay, that's the kind of DNA I'm talking about. I'm not talking about that. I'm sorry, that's so poorly drawn. So it's only in that linear DNA. Come on, you got this. Okay, and let me guess, erasing's not going to work. Nope. Okay, let's go to our next one here. And I'm just going to take us back to that page that we were at. Okay, let's get back here. Look how many of me are on this one page. That's kind of funny. Let me get rid of these documents. Maybe that will speed things up a little bit for us. Maybe, maybe not. Okay, so sorry, I'm literally 5 feet from my modem, but it's still pretty slow. Okay, so strand. On that lagging strand, we are going to have all of these primer events. Well, at one point, the primase will lay down a primer, which is fine. And then the DNA polymerase, so here I have that happening. This is the very end. Okay, this is the end of the sequence. And I have a primer here. And then I have a bunch of DNA polymerases I have a bunch of nucleotides that are being added with the DNA polymerase. Awesome. Here's the problem. A DNA molecule can't have RNA in it. So at the very end of this whole process, we know that these Okazaki fragments are created. And one of the things that has to happen is that RNA primer has to be removed. So I remove that RNA primer. Okay, great. I've removed that RNA primer. Here's the problem. DNA polymerase cannot add on any nucleotides because it needs, if it was going to start here, it would need to have a primer here. And it doesn't have that. It doesn't have anything to attach to. So that can't happen. And because it can't happen, the cell has to figure out what to do. Well, what the cell does is it actually cuts off those pieces that it was not able to make a matching copy to. Okay, so it just cuts it. It makes it shorter. What that means is you're going to end up, every time DNA replicates, you're going to end up with shorter and shorter linear DNA. Okay, the chromosomes are physically going to become shorter and shorter as they are replicated because of that lagging strand. You cannot, those last few bases will not be able to be replicated, and that's going to become a problem. So our body has some ways to deal with that. Limitations of the DNA polymerase create problems. Oh, wait, no, we already talked about that. Sorry, this is a new slideshow because I'm trying to get past that issue. So let's just look at this again. So this is just making larger what I was just showing you. And let me, let's see if I can do this. Do a laser pointer here. So this sequence here, DNA polymerase can't do anything with it. We took off the RNA primer. And DNA polymerase is like, well, what do you expect me to do? I have nothing to attach to here. And I have to work this way. Okay? I have to work from here to here. And I have nothing here. So what do you expect me to do? So because they can't do anything, the body says, okay, we're just going to get rid of this. We're going to eliminate this little piece here. Okay? Well, eukaryotic chromosomes have special nucleotide sequences to deal with this at the end of their DNA, at the end of their linear DNA. These sequences are called telomeres. Telomeres, I think, are really cool because they hold a lot of potential for things that we that are kind of, it's funny because a long time ago I read this book by Charles Sheffield. I've actually downloaded it as a book on tape or audiobook, and I think I'm going to reread it because it was really cool. In that it dealt with people. Well, I'm kind of getting ahead of myself, but it dealt with this whole telomere idea. And at the time, I didn't know anything about telomeres, and I thought, wow, that's really cool, but it's science fiction, right? Well, it turns out that maybe it's not. Maybe it's not science fiction after all. So you have these little areas called telomeres at the end of the DNA. Now, these telomere molecules are just basically DNA molecules, nucleotides, that have been added on to the end of the DNA molecule, and they do not postpone the erosion of the genes. But they said they do postpone. So what they're doing is that we are aware of right now, and that might change, they don't have any function other than to be cut off. So they, because they are being cut off at the end of each one of these DNA replication cycles, and because they don't, that we know of anyway, and I say right now because that might change, that we know of, they don't have a function in our body, they basically postpone any good genes near the end of the DNA from being eliminated. Okay, so anything that might, serve a vital function at the end of our chromosome could be affected by that shortening of the DNA each DNA replication cycle. But if you've got these telomere regions and you're just cutting off the telomeres won't be affected because that we know of, there is no function for these outside of to be cut off by the cell as it's going through replication. So one of the things that has been proposed is that telomeres, the shortening of telomeres, and I don't know if they can actually do this at this point or not, but we think that as these telomeres shorten, they're connected to aging. So in essence, a cell knows how old it is based upon how many telomeres it has left, right? The more DNA replication cycles it goes through tells the body this is an older cell or this is a younger cell, depending on how many times it's gone through that replication phase and how many times those telomeres or how short that telomere region is. Okay, so a cell can tell, or the body can tell how old a particular cell is. So that's kind of cool. If chromosomes, so that's the one thing. So the thing that was in my book that I thought was really amazing was this idea that if you could keep these telomeres from shortening, you could, in theory, as cells get older, they know, and we don't actually know necessarily how they know this, but they somehow know that, I'm old, this cell is past, I'm past my time, and I should commit, remember that word apoptosis? Looks like now my pointer doesn't want to change back. Oh, I know why. Let me get back over to the pointer here. Let me change this out here. I don't want laser pointer. I want pen. That's out, I think. Is that right? Yeah, there we go. So apoptosis. Sorry, wrong button. Apoptosis. So this is programmed cell death. Well, how is it programmed? We think that programming is related to the length of the telomere. The telomere gets shorter, and that cell will eventually commit apoptosis. It will kill itself because it knows that it's reached the end of its cellular life. Well, that then is related to aging. So if we could keep that apoptosis from happening, we could theoretically stop cells from breaking down and the body from aging. Okay, so So things like, getting wrinkles, and having organs start to shut down because the cells are just too old, and the cells say to themselves, I'm past my lifespan, and my liver is going to shut down. You can't live without a liver. Could we stop that aging of those cells by stopping the telomeres from, if we keep them from being cleaved off, would the body think, okay, the liver's fine, and it would just keep going? So there's some pretty cool research going on in that area, and we'll wait and see what happens with that. But on another front, and my sci-fi book actually addressed this. If germline cells, so germ cells are the cells that they're kind of like what's the word I want? Generic. They're generic cells. So think generic for germ, that can go on to become any kind of cell. Okay, so these are precursor cells that could become any type of cell. They could become a liver cell, they could become a lung cell, a blood cell, etc. But they will periodically divide, because as germ cells are needed, a germ cell has to go through mitosis, and as it goes through mitosis, you produce one cell that is a germline cell. So you start with a germline cell at the front, or at the beginning, and then remember, through mitosis, we're going to make two new copies. Well, one will continue to stay a germline cell, so that's how you always have a bank of cells left over. And then maybe this is going to become a red blood cell. Okay, so we'll make that a red blood cell, and it'll go on and become part of the blood. But these germline cells will stay germline cells from generation to generation. So then you do it again. Maybe now you make a liver cell, and that liver cell goes on and becomes part of the liver, but this germline cell stays. The problem is, each time you do this 1, 2, and 3 times, those telomeres are going to get smaller and smaller and shorter and shorter. And so eventually what will happen is you will end up with cells that are so short that the germline cells themselves will self-destruct. And then you get to the point where you talk about this with aging, you talk about the idea that now people can no longer, they have much harder time recovering from a surgery, or from an injury, or even just from a workout, right? It takes me longer to recover from a workout than it does for younger people. And part of that might be because of this whole germline idea. These telomeres, these cells are getting shorter, and they are getting eliminated. So there is an enzyme in our body that actually just exists. It's there. And its job is to keep those telomeres from shortening as the germline cells go through these DNA replication and cell cycle divisions. And that's called telomerase. So telomerase does what you think it does. It catalyzes the lengthening of telomeres, so it keeps those telomeres from getting shorter. It actually adds telomeres back onto them, so that those germline cells are not affected by this whole aging process. So that's good in theory. The problem is that we find that's not what I wanted. We find that, let me go to a new one here because we kind of messed that one up. There we go. We find that in cases of cancer, sorry, I got my pointer in my mouth. Give me a second here. In the cases of cancer, we find that there's evidence of elevated telomerase activity. So what might be happening is, normally the body lets these telomeres get shorter because, we know that as cells age, more and more of these errors will collect, and it's going to become more and more likely over time that those errors will lead to cancer. So by shortening the telomeres, it's a way of protecting the cells from cancer, because it only lets the cells divide for a certain number of times. So there's only a limited number of cell divisions before finally apoptosis takes over and says, that's it, I'm out. So it's good for those telomeres to shorten, long story short. But we still need the telomerase activity to happen for the germline cells so they don't get too short. But now there's evidence that suggests, growing evidence that suggests that telomerase activity is elevated in people who have cancer, or in cancerous cells, suggesting that what's happening is these cancerous cells, which should be targeted for destruction, are not, because what's happening is this enzyme telomerase is working to add on the telomere region. And by doing that, the body, the cell, is tricked into thinking, yeah, it's fine. It's of the right age. We don't need to get rid of it. We don't need to do apoptosis. And that allows the cancer to persist in those particular cells. So I think that's really cool. The book I was reading by Sheffield talked about how it was basically I like apocalyptic books, and it was basically about some type of solar flare that took out um, lots of communication and electronics and stuff. And there were these group of cancer people who were being treated with a drug that inhibited this telomerase activity. And they had to travel to, a couple hundred miles to get this, to try and, I think they had to find the researcher who produced this drug that inhibited the telomerase activity, because now the drug was no longer being manufactured because the world was going to heck, and they had to do something about that. And so, but they still needed their drug, right, or they're going to die from cancer. So they start on this trek of 200 miles, which wouldn't normally be a lot, but all the roadways are down, communications are down, electricity's down, all those things. Gas stations are down, all those things. So anyway, the kind of the gist of the story was these people's trek to try and get to this location to find the scientist who was working with the telomeres. Anyways, it's kind of a cool book. Charles Sheffield is a uh, physicist, and I do recommend his books if you like sci-fi, because he's what we would call hard sci-fi, meaning that he really takes he takes what's known about science and applies it to make it sci-fi. But the cool thing is, it's oftentimes not far from what we may actually at some point be able to do, which I think is really cool. All right, let me just see where we're at right now. See if I want to go any farther while these guys do their job uploading. Okay, yeah, I am going to go on and talk about 16.3 a little bit. We've got some time, and I would rather, especially if we might have a day off on Tuesday, I would rather get through this so that we only have that biotech stuff to do, rather than stopping early. I know you probably all are not exactly happy about that, but let's get through this. So 16.3 is talking, kind of continuing on about the whole regulation of, in a way, I guess, about DNA regulation, whether or not DNA gets turned on or off. So let's talk about this. Is just some general information about. Sorry, wrong place. So we know that bacterial chromosomes are double-stranded and they are circular, but there's not a lot of protein associated with them. They're a pretty small amount of protein. I'm going to switch over to just using the laser pointer so that I don't mess up the screen. So there's not much. There's a small amount of protein associated with double-stranded DNA that is circular in prokaryotes. In contrast, eukaryotic chromosomes have a large amount of protein. Okay, large amount of protein. So we're going to focus on those eukaryotes right now and talk a little bit more about that. In eukaryotic cells, DNA is combined with protein, and it's combined with protein into what we call chromatin. Okay, those proteins are called histones, and those histones are what is going to be responsible for allowing us to package a large amount of DNA into a small area. So I'm going to have a little ball here. Let's make this. That is my histone protein. It is a globular, or basically sphere protein. I'll make it a little bigger, maybe. There we go. I don't know why it's not. Man. Sometimes this computer. Anyway, so that's my histone protein, and this is my DNA. And the DNA is going to coil around that histone protein several times. Okay. Together, that represents chromatin. That allows the body, and then each one of these little chromatin packages will also get supercoiled into an even tighter ball. And that allows us to package a very large amount of DNA into a very small area, which we know is the nucleus. This next slide is a little bit more than we really need. It's just talking more about the wrapping around and how each one of those chromatin histones with the DNA associated with it will form into a bunch of beads that are called nucleosomes. But it's really more information in there than we need. What we need to talk about is how that chromatin is packed in the nucleus. So most chromatin is loosely packed in the nucleus and during interphase and condenses prior to mitosis. And that's when we go from being this kind of long and strung out DNA to being coiled up very tightly into what we know as an actual chromosome. So it's going to be coiled into that chromosome right before mitosis happens. Loosely packed chromatin is called euchromatin. During interphase, a few chromatin areas are highly condensed into what we call heterochromatin. Really, really densely packed. And that dense packing makes it really hard to express genetic, and that last word, sorry, is gone there, information. Okay. Now, we haven't gotten to protein synthesis or gene expression, but basically what that is talking about is the idea that DNA is a recipe, and that DNA will serve as a recipe for producing proteins. And those proteins are going to be made in the ribosomes. Now, that is something we will get to in the next unit. But sometimes we want to turn off these particular chromosomes. We don't, sorry, not chromosomes, these particular genes. Sometimes we don't want those pieces, those sections of genes to be expressed. We don't want them to make proteins. So histones, the proteins that the DNA is wrapping around, can undergo chemical modifications, and they can result in changes in chromatin condensation, and that can have effects on gene expression, and that can actually cause the genes to be easier to express. So it's easier for the proteins to be made, easier for those recipes to be expressed or be produced, or it can make it harder. Okay, so I'm gonna end on that, and then on Tuesday, because I think we're about done on time and probably full up on understanding right now. I need a little bit of time to process, but I will come back and I will talk about what that means in terms of gene expression. If you wind the DNA very tightly or you loosen it up, we have kind of a fun activity called Lick Your Rats, that is basically on nurturing. In other words, they looked at rat pups that were nurtured by their rat moms versus rat pups that did not get that nurturing behavior from their moms, and how their actual DNA, so something in their environment, their mom's paying attention to them, basically altered what happened in terms of their DNA winding and their ability of those genes to actually be expressed. So it's another kind of cool thing that we'll look at on Tuesday. So for now, that is all. Please work on your study guide. Continue through. I don't know if I can find where we're at here in terms of the study guide. Let's see if we can find it. There's our study guide. It looks like my neighbor is snow plowing my sidewalk. Yay me. Means I don't have to. I get this to load. And I think I had said get through 36. Now it looks like you can get through 39. Didn't get very far. So you don't have a lot to do. There's just some stuff on mismatch repair, mutagen. I don't know if I talked about mutagens specifically. Mutagens are just anything that can produce a change in the DNA sequence, because a mutation is a change in the DNA sequence. Okay. So go through 39, and then this biotech here. We just have a little bit of biotech that we're going to do for next week, and then we'll have the test on Friday. So as always, if you've got a question, please email me, and I should be around all day, and I'll get back to you as soon as I can. Have a great weekend.